everybody for coming today. Our special and honored guest is my good friend and colleague, Dr. Jimmy Bagley. Dr. Bagley did his undergrad in California at Cal Poly San Luis Obispo. He then did a master's degree under the tutelage of Dr. Judelson at California State Fullerton. And then he did his PhD in the Ball State Human Performance Lab. So when I came into the Ball State Human Performance Lab, Jimmy was just about through, and he basically spent 2 years teaching me to write and to do science. So I owe him tremendously for that. He's now an associate professor in his 6th year, correct?
Yes.
But I can still remember things better. In his 6th year at San Francisco State University, where he's the director of the Muscle Physiology Laboratory. For those of you who want to reach out to him after the presentation, there's his email, and you can follow him on Twitter and Instagram. So without further delay, welcome, Dr. Bagley. Given the nature of his presentation, we talked about this earlier, it's kind of broken down into 3 parts, and we think it'll flow better if we have you guys ask questions following each of the individual sections. So he's going to kind of present this in thirds, and then there'll be an opportunity for questions and answers as we go through that. So,
Well, all right. Great. Thanks, Greg. Thanks, everybody, for having me. I appreciate it. We're just chatting. This is my 2nd time speaking in front of a live audience in a year and a half. So it's been pretty weird. And I'm just happy to be able to have the opportunity to talk to you all. So thanks again. Again, I am from San Francisco State University. This is our campus in the beautiful southwest corner of San Francisco. Been there about 6 years now. I run the muscle physiology lab. I also work in the exercise physiology lab and strength and conditioning lab. So kind of share a little bit about that with you. We do have brand new ex-phys labs with the teaching lab. We do a lot of our applied research in there. We do applied research in our strength and conditioning lab. And then some of the basic research in our muscle physiology lab. So we've been spending the last few years building that. And if any of this entices you, maybe I can get you out of Georgia if you'd like to come to our grad program. There's a link right there.
we'll trim that out of the presentation.
No, you guys have a great program here. It's been really cool to hang out for a few days and check out some of the research you're doing. So I've actually learned some stuff here too as well. So thanks again. So basically the way that I see physiology, I'll kind of do a little intro here before we really get into it. It's kind of an integrated view. So I look at it really from top to bottom, bottom to top, from the whole body all the way down to the molecular level and everything in between. So when you think about whole body, you think about what you do in the lab over here in your applied lab, things like, weightlifting is a whole body maneuver, or something like a VO2 max testing, something like that, where you can get data on many organ systems and many cell types working together. We take it the next level down, right? And then we're down at the organ systems level. So this is when you take like renal physiology, cardiovascular physiology, muscle physiology, and so on. And you build those blocks together. The next level below that we see is the cellular level. So this is an example of a muscle cell. These are muscle fibers, are actually my muscle fibers. We'll talk about that's what kind of got me into exercise physiology in the first place. I was just interested in how my body worked, probably like some of you. So these are each individual muscle cells. And then from there, I'm going to show some pretty cool images from our cell imaging lab to show you what we can do with these. So this is a muscle fiber. You remember from class, you have multiple nuclei on there. So muscles are syncytial, meaning it's not just one nucleus, it's going to be multiple nuclei in there. And then from there, we can get down even to the molecular level, so smaller, down in the protein and gene level. And then I'll even talk a little bit about epigenetics and the microbiome. So I don't know how deep the rabbit hole goes. I don't think we're down to the bottom yet. It's just going to keep getting more and more kind of intense down there. So I'm going to talk a little bit about that. And then again, how all this works together to influence human health and performance, and then also adding technology. So I don't know if you saw the title of my talk, but basically I'm going to talk about technology use in exercise and exercise physiology research. I'm not going to talk about fitness tracking and that kind of stuff. I'm going to talk about a little different technologies. This virtual reality headset. Okay, so I've always been interested in technology. I'm not going to read this to you, but this was my first grant proposal at age 11 to my father. So I wrote a grant to my dad. So this was like mid-90s, and laptops were like the new thing. I watched Jurassic Park, and I was like, man, I want a laptop. I didn't really know what they could do or whatever. I'm not going to read this, but again, I did get that laptop, and that's a Toshiba Satellite Pro right there. So that was top of the line. And that was my first real piece of quote unquote technology. Something that I use, and I was really privileged and fortunate to be able to get that, because I definitely wouldn't be talking to you today if I didn't have access to some of this technology early in my life. So what is technology? Just textbook definition, application of scientific knowledge for practical purposes. So anything could be technology. It could be, this laser pointer, this pen is a form of technology. Everything around you is some form of technology. And when you think about it, you can think about new technology, which would be something like an emerging technology, something brand new, and then converging technologies are things that come together to create something new. This is happening all the time, faster and faster. So without giving a history lesson back to 1400, we're just going to kind of start with one of the biggest things to start off this technology boom, kind of back in the beginning of the Age of Enlightenment, or whatever you want to call it from a historical standpoint, with the printing press. Why was the printing press more important than writing things down with a pen? Mass produce information, get it out to people quickly and efficiently. So that kind of kicked us off, and then where we are now. Let's just keep going. Telescopes, microscopes, cars, airplanes, and technology is getting more and more. It's kind of growing more and more fast each year. I mean, I don't even know what the next thing is going to be, but it's going to keep going. And I'm going to talk about a few of these as we get in here. Okay, so technology is a double-edged sword. I'm going to give you a rosy picture of all the good things you can do with technology. But you have to remember, technology might not always be the right answer for what you're trying to do. So this is, just kind of one caveat slide. Things to consider when you're thinking about technology are things like equity and accessibility, right? Technology is often very expensive. And again, I was really privileged and lucky to be able to get access to some tech as a kid. Also, think about cost and things like that. So as technology is around longer, it will become more and more affordable. Other things, use and overuse and abuse. Too much technology could be a bad thing. If you're always looking at your watch and you're always thinking like, oh, my heart rate variability is so messed up, that might stress you out more, and then it'll mess up even more. So think about, there's a time and place for technology. You might not want to use it all the time. And then also future ramifications. This is something you can't predict. There's kind of known unknowns, and then there's unknown unknowns. I'm sure when they invented a car, they didn't think that was going to increase the obesity epidemic. There wasn't any of that going on before cars were invented. So think about, cars are easy to get around, but then we're not walking or riding or whatever. If you're interested in more of these kind of downsides of technology, check out this book. It's our colleague, Dr. Andy Galpin, who's another Ball State grad down at Cal State Fullerton. It's called Unplug. It's not anti-technology, it just makes you think about some of these things. So I would recommend that if you're interested in checking that out. There's also, you don't want to read the whole book, there's a couple of good YouTube videos you can check out to see that. Okay, so what's the plan for the rest of the lecture? Like Dr. Grosicki said, I've broken this up into 3 parts. So first thing we're going to do is look inside the body, inside the muscle cells specifically, because I was trained as a muscle physiologist. That's what I know and what I really like. And then we're going to look outside, get into the microbiome, some of the stuff that you're all doing here at Georgia Southern. That's pretty cool stuff. So the microbiome or microbiota are all the things that live on and inside you and around you that can actually influence your health and your performance. And then we're going to look forward, maybe into the future, not so distant future, I think. And looking at things like the future of fitness, I'm going to talk about some of the work we've been doing on virtual reality. So after each one of these, I'll open it up for questions, and then we'll probably have time for questions at the end. So this always happens at the end, but I'm going to show it now so we don't forget. But a lot of this research couldn't be done, or all of it couldn't be done without our funding and support. Just full disclosure, we've had some stuff donated from Nordic Track, Protein Simple, Virtual Reality Health Institute, and then these are our funding organizations that have helped make this possible. Then I want to also, before we start, acknowledge my students, because I'm the one that gets to get up here and talk about all this stuff, and they're the ones back in the lab, like you all doing the work. So I get to talk about all the cool stuff we're doing, and then all the collaborators around the country that have made all this happen as well. All right, so let's get into it. Inside the muscle cell. So remember, I started as a muscle physiologist, really at Ball State with Dr. Grosicki, isolating muscle fibers all day. Ended up paying off because I learned a little bit of something about muscle fibers at that time. So, basically when we talk about muscle, I'm going to talk about 3 components of this as well. 3 is my favorite number. If you can't tell, everything's in 3. So you got metabolism, which is kind of the foundation muscle, right? You've got to make energy and use energy. You're doing it right now, even though it doesn't seem like it, you're still using that energy. Hypertrophy is muscle growth, atrophy, muscle loss. So these are all kind of connected together. We'll talk about some of the research I've been involved with each one. And here's that cool picture of a muscle fiber. You can tell these little blue kind of dots, those are called muscle nuclei. And we can even get tighter in there. Each nucleus in your cell, so your muscle fibers have hundreds, probably thousands of nuclei in each one. Each nucleus has a full copy of your DNA, which is about a meter long, and that's wrapped into a 15 micrometer long little, segment of a organelle. Pretty amazing stuff. But all these pictures were taken in the Cell Molecular Imaging Center. It's in our biology department. We have pretty good collaborators there. We're able to send students over there to do all the imaging and bring us data back. So how do we get these pictures? We've got to, obviously use some technology. These pictures, you can't just take a picture with your phone or whatever. These are really, really high resolution images. And what they require is if inside your body, there's no color on your cells, they're all clear, everything's see-through. There's no need for there to be any blues and reds or anything that you can see. What we've got to do is label these, and we label them with little fluorescent dyes. So a little fluorescent thing like a glow stick, basically. When you hit it with a certain wavelength of light, they'll bounce back different wavelengths of light. But we're not using just a normal microscope, we're using what's called a confocal microscope. And the idea of this was discovered or invented in the mid-50s by a guy named Marvin Minsky. So look up that guy if you're interested. He also invented the pacemaker, I believe a few other pretty crazy inventions throughout the time. But confocal didn't get really popular until the 90s because that's when computing power got really, actually powerful enough to be able to get these images. Okay, so first we'll talk a little bit about metabolism and basically a little study we did with Cal State Fullerton on AMPK, which is, I would call it the cell's energy sensors. Anybody learn about AMPK in class at all? Maybe in biochemistry or exercise physiology? Okay, cool. So you kind of have an idea of what it is, but basically what we did was we looked at exercise perturbation, high intensity interval training. Anybody's ever done this, you use a lot of energy in a real short amount of time, right? And what that does inside your cell is it decreases the amount of ATP, the amount of energy you have, and increases AMP. So that ratio changes, and that turns on this little protein called AMPK, or AMP activated protein kinase. And then that in turn affects downstream targets. And a few targets we looked at in this study were ACC, TBC1D1, and TBC1D4. You don't necessarily need to know exactly what those are. Just know that AMPK, once it's activated, it phosphorylates these or turns them on, gets them ready to work to basically help break down free fatty acids. So that's a good energy source in your cell, right? You got fat in there. Also bring glucose into the cell. So you've got glucose transporters in your cell membrane. You can bring those in, and these are two, one of our colleagues calls them the window of gains genes. So if when you lift, right, you've got to eat pretty quick. Well, not immediately, but you've got this window of gains, and these are some of the genes that are turned on, proteins that are turned on for that. Okay, so just a quick background on this study. Again, it's on high intensity interval training, and it was using a pretty elite athletes. We had MMA fighters and CrossFit athletes, both men and women, come into the lab and do 6 rounds of HIIT. I believe these was 90 seconds on, 60 seconds off, 100% of VO2 max. So pretty hard. By the end of it, I did this as well. I always do the studies, just like Dr. Gosik, and I think by round 3 or 4, I was in the trash can. Not doing so good, but we did this. We also added some biopsies in there. So we do muscle biopsy procedures at some of the Cal States. And what this entails is a small needle, about the size of a pencil, and it goes into your thigh. We go for the quadriceps, particularly the vastus lateralis. It's lateral, it's on the outside, not much vasculature, and you use it all the time for cycling or standing up or squatting or whatever. So we did 3 or 4 serial biopsies, 1 pre, 1 immediately post, 90 minutes and 180 minutes. This is what the biopsy needle looks like. We get a pretty big sample, about 100 milligrams, so like the size of a pea, maybe a little bit bigger even, and then process it. So this is me with much less hair a couple of years ago before the pandemic, and basically taking those samples and immediately throwing them in liquid nitrogen so we can flash freeze them, and then we have a time point, a frozen time point of what's going on with metabolism inside the cell. There, you can see Karen here is about to throw a water bottle with this guy. He didn't care about the biopsy at all. He was just so, gassed after the 6 rounds right here. So then with this, first thing we had to do was we had to see how we're going to look at AMPK. And if you're in, biology class, you've probably done Western blots or heard about Western blots. That's kind of the standard way to measure protein content or what type of protein you've got there. So we actually started working with a company called Protein Simple. We were looking at a new way to measure proteins called CNIA, capillary nano immunoassays. And what that is, it's basically, they call it protein simple because it's a simple Western blot. Just this one's called West, kind of the medium-sized one. There's also big ones, tiny ones. But what this does is it basically simplifies everything for you. You load your samples in, and then it automatically does everything. And instead of doing a blot, like you would with a Western blot, which I'll show in a second, it uses these little capillaries that are kind of pre-made and is able to tell the quantity and the type of proteins that are in there. This is just a short video. It's on their website if you want to check that out. But these things, you know, are becoming more ubiquitous around. I've seen a few at different labs now that they've been around for a few years. They seem to work pretty well. So what did we find? So basically, again, this was kind of our procedure. We took that biopsy, like I showed you, flash froze it, liquid nitrogen, preserve the muscle, and then isolating fibers. So this is a fun thing that my students get to do. They're probably doing it right now. Isolating single muscle fibers, very tedious. We dissolve those, and then we're able to fiber type those. So everybody know what fiber type is? You've got fast twitch and slow twitch fibers. You've also got some intermediate types. So 1 is a slow twitch, 2A is a fast twitch. You've got some kind of super fast twitch fibers in there as well, but 1s and 2As are your more common ones, along with those hybrids. What we'll do is combine those and isolate them. Sometimes we'll even do a single cell, which I'll show you here in a second. And then this is the data that comes out on this, this device to show you basically the content of those proteins. We can take ratios, kind of use a standard protein in there to take a ratio and show how those change over time. So what do we find? So this was the really cool thing with this study is we basically did a Western blot on AMPK beta 2, which is just one AMPK subunit. This is what the Western blot looks like when you take fibers per microliter down. So by here, by 0.02, it's unusable data. You wouldn't be able to use that in the lab. Then you can take a look at the CNAI, and you can see it goes all the way down, basically to the single cell level. And you can even get smaller, clip a little piece of the single cell off, and you can get data from that. So pretty cool technology to use for that. And with that, also, this is all of, I can't take credit. Dr. Irene Tobias, she's the one that came up with all these ideas and worked with Protein Simple. She's down at UC San Diego right now, and she was doing some COVID work too. So she's actually a biochemist, wannabe biochemist. So I get to talk about it from an exercise standpoint. But we did find there was fiber type specific differences in expression, which we kind of expected to see. And then she went on to do a little bit more data analysis on this and looked at the difference between men and women and between fiber types on this as well. So if you want more information on this, I'm not going to get too into it, but she went through all those proteins I talked about, and they just published this recently, just earlier last year. And what our lab is working on now is we've got those samples, and we're trying to take a look to see if we can corroborate that data with imaging techniques with our confocal microscope. So this is pre-high intensity interval training, and I've kind of zoomed in here. This is 2 nuclei. These red lines are actually actin, so you know your muscle fibers got actin and myosin, right? So you can actually see the striations in there. And all these little green dots, that is AMPK. We've always got it there. And this was at rest. This was a 0 time point at rest pre. This is an image from immediately post high intensity interval training. Just like we saw on that protein quantity analysis, we can see it light up. You can even see it co-localizing over and inside the nucleus, going in there to do its work. So we're trying to get more imaging data, and all this stuff is in preparation right now. Okay, so that's metabolism. That's some stuff. Metabolism we've been working on. We've also been doing things with muscle hypertrophy. Anybody ever heard of myonuclear domain in class? Not really. I'll kind of explain it a little bit. So this is something I got interested in about 7 or 8 years ago. I heard about it, and I was like, that sounds pretty cool. But basically, the idea is, you see this longitudinal fiber. If we were to turn that on its axis and you're looking at it like a tube, the nuclei hang around the outside. And the idea of myonuclear domain is that each nucleus controls a certain area of that cell. So in this case, you got 4 nuclei, each one gets a quarter. If the cell gets bigger or hypertrophies, like you lift weights, resistance training for 8, 12 weeks, something like that, the nuclei have got more space to take care of, and they might not be as efficient of sending signals out to that cell. So you'll have to add more nuclei. Your pizza slices go back to the normal size. So new nuclei have to be added for sustained hypertrophy. This has been the idea for a couple decades now. And obviously we're kind of getting a little bit more into the nitty-gritty of how this all works. So some people like to call this muscle memory. It's not really muscle memory, kind of muscle memory. Your muscle remembers how big it is, because what people think is you can have like a satellite cell, which is a stem cell, attaches to the muscle fiber, can add new nuclei. So this would be your first resistance training program, for example. Your muscle fiber gets bigger, you add more nuclei, right? Then let's say you detrain, something happens, you can't exercise, you get injured, so you're out for 5-6 months. Muscle fiber will get smaller, but potentially those nuclei hang around. They're just kind of waiting for another stimulus to come back. Then that secondary or subsequent training program, your muscle might be able to grow a little bit faster. And then if you needed to add more nuclei, you could do that cycle again to a certain point, right? Your muscles aren't going to keep getting bigger indefinitely. There'll still be a limit. So this all sounds good. And I believe this to a certain extent, but as always, it's more complicated than this, right? There's always going to be that next level down we get to explore. So some things to consider when thinking about this myonuclear domain hypothesis: people's age might be different as you get older. Little babies have a ton of these stem cells. They're able to grow nuclei like crazy. Once you get into your 50s, 60s, 70s, 80s, you lose stem cells and you lose the ability to do that. Also training status. If you're already really trained, you might already be at your ceiling. You might not be able to add anymore. Other organelles as well. So there's things like your myonuclei have all the DNA there. They send signals out to ribosomes, which hang out around there, and the ribosomes make the protein. So maybe it's more related to the ribosomes. Another idea. And then fiber type. So I showed there's differences in fiber type with different things. There's also differences in fiber type with nuclei. Now, if you're really interested in getting deeper into myonuclear domain, I would say you should look up Dr. Kevin Murak. Dr. Gosicki and I were hanging out with him out in Arkansas. He just started a new lab at University of Arkansas, and he's getting really deep into this work with mouse models, because they're able to change things. Humans, you can only do so much. We're not going to chop off somebody's leg and count every fiber in there. That'd be obviously unethical. But with mice, you can do all kinds of stuff, and they grow much faster. You can change their genotype and stuff like that too. So this just shows you've got a type 1 or slow twitch fiber, type 2, a fast twitch fiber. If you have satellite cells or these stem cells and you make them exercise, they will hypertrophy and they'll add more nuclei, just like you'd think. Well, his idea was, what if we take the satellite cells away? Are these even necessary for hypertrophy? So again, yes and no. They found out with type 1 fibers, without satellite cells, they were able to just remove all the satellite cells. They didn't see any hypertrophy after training, but with type 2 fibers, fast twitch, they still saw hypertrophy in the absence of satellite cells. So this throws a wrench into our idea that it's required to add nuclei or satellite cells to the muscle fiber to grow. So again, here's some cool pictures. These are cross sections of muscles just showing that. But if you're interested, check out some of Dr. Murak's papers from the last 5 or 6 years. He's been publishing a lot in this area. The one thing that we did in our lab was we do human research, mostly not animal research. And I had a student, this is Stephen Machek, and he's now at the University of the Ozarks. He's an assistant professor out there. I stopped there on the way as well to talk to his class. But he looked at muscle fiber type, morphology, the myonuclear domain in a middle-aged elite powerlifter using anabolic steroids. So there's another thing. What happens if you add steroids or some kind of exogenous hormones to a person or an animal? So this is a picture of one of that athlete's muscle fibers, and it's hard to compare it to other ones. You can't really see how big this is, but it's actually pretty big. We saw on average, fast twitch fibers of this elite powerlifter were not much different than an average human. I have some other animals to show you. Mouse fibers are real tiny, rhinoceros fibers, very large. A couple of his fibers, though, were 3 standard deviations outside of the average. Extremely huge. One of the biggest fibers I've ever seen of any animal was from this individual. It also had more nuclei than I've ever seen in any animal or human. So this is just kind of showing that, this is a case study. I thought it was a pretty cool case study because we had this case, but that there can be differences in things like myonuclear domain and hypertrophy, depending on what you do to that person or animal. And then this just shows the myonuclear domain. So you can imagine each nuclei takes care of less space. It could be more efficient. And this individual, much more efficient here. Average males, and then here's different age groups, and then an age match control for him too. So is this thing to do steroids and all that? No, not necessarily, because there's a lot of other things going on, but they did make some of the muscle fibers extremely large. And this is some of the reasons why. Okay, what about atrophy? So this is the third part of muscle that I'm going to talk about: spaceflight and unloading. So Last night, we were trying to catch a glimpse of SpaceX launch. We didn't see it, but if you were looking in the southeast sky, you might have seen a little speck go across. SpaceX launched 4 humans into space, and this is happening almost every month now. Every couple of months, they're sending humans into space. And you can imagine space is not where we're supposed to be. We're supposed to be on land. So when you go up there, things happen to your body. Some things that happen is you get reductions in your cardiovascular, bone, tendon, and muscle health. And this can be bad, especially if people are going to go to the International Space Station for months, Mars for years, or maybe forever. What's that going to do to their body? What can we do to help mitigate some of these things? Exercise right now and some nutrition, some of the only things you can do while you're in space. And even, this was up till 2010, so only about 11 years ago. But despite all their best countermeasures, they still saw significant decreases in muscle mass, performance, and a shift in fiber types. I'll share some data here in a second. This would be not good if you had to do something like an emergency egress, get out of the spaceship fast, and your muscles are 35% weaker than they were on Earth. It's not going to be good. Or if you have to land on a planet or come back to Earth, it's not going to be good. So kind of following up on that fiber type shift thing, this is a study that I did. It was just a review article from 2012, kind of showing all up to that point, all of the unloading studies and what happens to your fiber type from slow twitch, fast twitch, super fast twitch, and those intermediary types. Basically, we saw across the board a decrease in slow twitch type. So your body's starting to get rid of these metabolically active fibers and kind of converting them into these hybrid type fibers. So we saw a huge increase in hybrid fibers across the board. This, along with the atrophy that you see, is pretty bad. And you can come out of it, but it might take 6 months or a year to get back to where you were, depending on how long you've been in space or unloaded. What are they doing now? For the last few years, they've been doing this integrative resistance and aerobic training study. So if all of you were going to try to maintain your muscles, what would you do? You probably wouldn't go, train for a marathon or a 100-mile bike ride. You probably would lift weights, maybe do high intensity interval training. So, what these studies have been doing is looking at space as well as bed rest. So this is the head down tilt bed rest model. So imagine laying in bed for 30, 40 days or a few months with your head tilted down to mimic spaceflight fluid shifts, and you can't get up to go to the bathroom, anything. So NASA will pay you $20,000 to do this, but I don't know if I would do it for $20,000 unless I knew I was in the exercise group at a randomized. But anyway, what they're doing is they're looking at high intensity intervals. So this is their kind of 6-day training plan. The resistance training is new, relatively new in space. They've been doing continuous aerobic training for a while, and then the interval training is new. So they're trying to do more high intensity resistance training up there to maintain these astronauts' health. And if you're wondering how they do that in space, as you can imagine, weights, you'd be able to basically take your 300-pound dumbbell and just toss it up in the air, and it would just keep going. So they've got these devices that this almost looks like a clamshell. This is called the ARAD, and it folds up, and it's able to load 300 pounds on there using pneumatic devices. They've also got a bike in there, and then a treadmill that you can strap in. The almost one gravity, it's pretty close to it. You can imagine that it would still be relatively uncomfortable. But you can do pretty much everything you can do in the gym in space now. So, with that, kind of our lab is starting to look at things. I was saying we don't do a lot of animal research, but we just got some tissue from NASA to look at some muscle of mice that have been up to space for several weeks. And then we're looking at their tendons as well to see how those change over time. Anybody have any questions on inside the muscle cell or any of the muscle research? Yeah.
Have you seen any differences with myonuclear domain depending on what type of hypertrophy? Because I know there's like the functional where you actually have the like the myosin and actin chain getting thicker or more versus like just sarcoplasmic hypertrophy.
Yeah, so we haven't looked at that specifically. That's a good question. So sarcoplasmic hypertrophy people talk about a lot, but like you said, that's the fluid changes really in there, not the actin myosin. I think what you'll see is sustained hypertrophy with actin and myosin increases. You're going to probably need to add more nuclei. Well, that one study shows maybe not in type 2 fibers, right? So might depend, but yeah, that's a good point. There are different types of hypertrophy. Most of the hypertrophy I'm talking about is myofibrillar. Adding actin myosin requires making new proteins. It takes about a month or so. I don't know, if you started lifting now and you hadn't been lifting, you might see gains in 6, 8, 12 weeks, somewhere around there. Good question.
With your original HIIT training, where you did that 6 rounds at AMPK before and after, did you notice any difference in pre and post AMPK for type 1 muscle fibers versus type 2 muscle fibers?
Yeah, there's differences just at baseline. Yeah. Yeah, so I think I showed in that figure that type 2 has more AMPK, but it might be phosphorylated less. So activation is going to be different as well. So you might have more, but it might not be activated. Yeah, that's a good question too.
Would the high intensity interval training stimulus, you think it would provoke a greater interference effect than a more lower intensity steady state based on the magnitude of the AMPK response?
That is a good point as well. So like interference with resistance training, you mean?
Yeah.
Yeah, I think it would all depend on the total volume. If you can volume match the, the hit and the continuous might be similar, but yeah, that is a good point. So with the interference, and it might also depend on how close those exercise bouts are together too. But yeah, with this, we just had them do that one and they, I think it's 72 hours before they weren't allowed to do anything. And then we didn't really look into that, but that is a good point. I know that concurrent training is getting a lot of press lately. People are quite pumped up. Yeah.
So I don't know if you're going to get to this, but since you talked about hypertrophy and things, have you been looking or have you looked at mammalian target of rapamycin?
Yeah, so mTOR was also in that study that Irene did, that ran it. That was one of the targets that she looked at as well. But I know that AMPK and mTOR kind of have converging roles in there, right? And so with that study, I think that mTOR did change over those immediately post up till 180 minutes.
Because am I correct? I remember that typically when you have situations where AMP kinase is activated more, indicating more fuel substrate supply and things like that, that tends to, they do tend to be inversely related.
Yeah, so, and I think she did show that in the figure up the look. That paper has a lot of figures in there, so I have to dig that out. But I'm sure that went down, but then, over time that might start ramping up again. If we didn't get past 180 minutes, that would probably be something that would be 8 hours, 12 hours. At Ball State, we did 24-hour repeated biopsies. I don't have the time, energy, or money to do that, but I would love to do that. Yeah, that's a good point, though. Is there a question in the back too?
Yeah, I was gonna ask for non-nuclear domain, is there any research done on potential benefits or just performance in terms of
Like how many total satellite cells per unit of muscle, I guess?
that or just getting that difference between certain activities and how
Yeah, I think that hits on the point of aging, because that would be the only model in humans we have of looking at fewer satellite cells. And it's hard to count too. You can count, in an average adult, whatever that is nowadays, it's about 5% of all your nuclei are marked as satellite cells. So it's not that much. Kids, again, have a lot, 2, 3, 4 times that. Once you get to be 70 or 80, you might only have 1% of your cells or stem cells. So that's going to change basically how fast you can adapt. You might still adapt, but I think it's more of the rate. Each satellite cell can only be one place at a time, right? But yeah, that's a good point. That's kind of a model. In animals, you can turn those on and off and ramp them up, but in humans, it's hard to do that.
With atrophy and spaceflight, would you expect muscle fibers to atrophy quicker than ligaments and tendons, or vice versa?
Yeah, muscle will always get bigger or smaller
Yeah.
faster, and the ligaments or tendons should be slower. And that's what we're kind of looking at, the time course of tendon morphology, because most injuries people come back are going to be soft tissue, right?
Yeah.
Tendons and ligaments. They take longer to build up, and they take longer to break down as well. That's a good point. I think if you're interested in getting into tendons, that's kind of an open field, especially with unloading and aging.
Yeah.
Everybody focuses on muscles, the most important, but it's got to be connected to your bones.
Exactly.
Yeah, good point.
I was just wondering if you were aware of any data, and obviously maybe with aerobic exercise in my nuclear domain and how that may influence it.
I haven't looked into a lot of that because, everybody's looking at hypertrophy and stuff, but I'm sure it would, but maybe those type 2 fibers are the ones that, you're turning on your type 1, type 2, all of them when you're aerobically training, depending on what you're doing. So I imagine it still would impact that, but that's a good point. I haven't looked into that too much. Cool. Yeah.
I know there's not really research on it in humans, but with the myonuclear domain, you have the increased size of the fiber and then you have more nuclei. Do the fibers then split to increase the number of fibers and then those increase? Or is that something that's been investigated in animal models?
Yeah, that's another idea. So there's the idea of hyperplasia is adding new fibers, either from its own state of satellite cell, it could actually become a myotube, it could actually become a fiber, or in animals, Dr. Murek's shown that fibers will split up in half and become two separate fibers. So it does happen. And I think in humans it happens too. It just probably is not as significant as your myofibrillar or sarcoplasmic hypertrophy. Maybe like 2% of your gains or whatever. I don't know a number would be related to fiber splitting. But saying that, that individual that we had that was the elite powerlifter, he had a lot of fibers. And if you notice, they weren't that big. He had quite a few fibers total. So we couldn't again count how many fibers he has compared to everybody else. But it seems like maybe that was the case with anabolic steroid use, resistance training. Those might cause things like the fiber splitting. That's a whole another research. You guys both go into the tendons of fiber splitting and you're set for the rest of your life.
We train them well.
Anything else before we jump into microbiome? I'll take questions at the end too. Yeah.
Well, I don't want to, I'm sorry. So, and I'm.
You better be sorry, Tom.
Expressing my ignorance here, but I mean, it's myosin heavy chain. I guess that's still sort of the go-to in terms of fiber typing. I know there's, there's APAs and a variety of others, but my limited understanding is that myosin heavy chain, although it correlates very well with things like twitch, like Vmax, twitch velocity, things like that, that it often does not correlate very well with preferred bioenergetic enzyme content, and therefore metabolics. Is that, am I incorrect about that, or is that they can really vary independently?
You're right. So if you look at it, type 1 and type 2 on average, like you're saying myosin heavy chain will have different amounts of AMPK and different, citrate synthase and all these other enzymes. But myosin heavy chain 2 fibers have such a huge range. You can have really oxidative 2 fibers, really glycolytic 2 fibers. So, if you go back to the 70s and 80s and Dr. Reggie Edgerton, some of the other Ken Baldwin, we started doing this work in animals, they were like, we need to type it based on a protein content for twitch, plus also an enzyme. That would be ideal. But again, how many, it'd be like, oh, we should measure body comp using the DEXA and BOD and at some point you got to pick one. I think the community in about 2010, 11 picked myosin heavy chain. But, now there's other ways to do it. Maybe there's different genes we could do. We don't even maybe need to do heavy chain. And it's not just 5 types, it's a continuum. So there's thousands of types. That's a good point. No, I'm trying to write a paper on that, the historical perspective.
It's always very easy, especially when we're teaching these classes, to try to make it sound like they're all unified, but in fact they're not. They're all different types, different methods, and we have to remind students sometimes, as well as ourselves, that you have to sort of stick with the type and not try to mix them.
Right.
Mix the discussions about them, if that makes sense.
We like to bin things, humans will bin things in these categories, but it's really a continuum.
Yeah.
It's great.
Thank you. In that elite powerlifter, did you by any chance notice it is like type 1 fibers, which is typically a lot larger than average type 1 fibers? Because I know there's sort of like, it's still just a theory and not even like a theory that's been tested, just like ideas people have that for like the really elite level bodybuilders, you get like much like inhumanly large. The question is like whether or not they've just found a way to really hypertrophy their type 1 fibers greater than others. Because I think like the big example is like Ronnie Coleman, which is like such a freak. And I know anabolic steroids are of course larger, but what makes someone have the ability to get so much larger than even other people in anabolics?
Yeah, well, I didn't show the fiber tech data, but he only had like 10% type 1, pure type 1 fiber. It was more of 60, 70% 2As and then some of the hybrid types as well. But maybe you're just able to get more 2A fibers that are oxidative or that are oxidative. Like if I could have all my fibers type 1, I would have type 2 fibers, which it could be oxidative or glycolytic and still powerful. So we didn't show a lot of data on the ones because we didn't have a lot. We pulled a lot, pulled hundreds. I think we pulled almost 1,000 fibers out of there and only found about 10% pure type 1s. Wow. So maybe he shouldn't be running marathons and stuff. Cool. Let's jump into the next section. So again, I started my career looking into the body, and then now that not that long in my career, 6, 7 years in, I started looking out. Wow, everything around you, your space flight, your environment affects what you eat, affects your performance. And so I started looking at the gut microbiome. Actually, with Dr. Grosicki, he helps me, kind of spur some of these ideas with our students we worked on, but gut microbiome is really the genome of all the critters that live on and around you. Gut microbiota is the name of them, actually. So I'm going to kind of use those interchangeably, but the microbiome is actually the genome of them. We're talking about bacteria and viruses and everything. Specifically, we're going to talk about the bacteria in the GI tract. So if you're interested in this type of stuff, the Human Microbiome Project's been going on for a while now. They started publishing their first data almost a decade ago. They've got a ton of information and it's still being looked through. So this is an ongoing thing. What they know is, you got about 39 trillion or more bacterial cells in your body. This equates to pounds. I don't know what the number of pounds is now, 5 pounds in your gut specifically. So that's how much bacteria you got living in there. And this is essential for your life. This changes throughout your lifespan, changes in things like diet, and now we know exercise, and it can be a contributor to human health and disease. And they've said specific types of cancer, cardiovascular disease, obesity, diabetes, dementia, and maybe even aging itself can be related to these bacterial cells that live inside of you. I'm going to share a few animal studies to kind of show you how important this is. So I'm not going to get too much into the animal data, but what they did, this was done again not too long ago, a little over a decade ago, was the first one, but they took a group of mice, they fed them a high-fat, high-sugar Western diet, and of course the mice got obese, right? If you eat too many calories, especially these types of really dense calories, it can cause obesity. That also changed their microbiome composition. Well, what they can do is actually take germ-free animals. This is another thing you can't do in humans. You don't want to take all your germs out, but not good. But they have these mice that they grew up in a clean environment with no germs in their body or around their body. They fed them that exact same diet. They had no change in phenotype, so they didn't get obese. Obviously, their microbiome didn't change. So this shows, the gut bacteria might be the thing that's causing this. Of course, this is kind of just observational, so they had to do a few more studies. This study, what they did was they took 3 groups of those germ-free mice that have no germs in them, and then they had basically a microbiota transfer, so a fecal transfer into the gut of these mice, where they put normal, quote unquote normal, obese, and underweight bacteria into them, fed them the same diet, and what happened to their phenotype? They changed to the phenotype of the mice that those came from. So a normal one, an obese one, and an underweight one. So the gut bacteria, independent of food, changed their body composition, body type. So that led people to think, what is this? What did the bacteria do in humans? So this was a pretty good review paper that came out. All this stuff Dr. Garcia can share with me. So I'm basically, hopefully I'm not repeating everything you heard in your class already, but this is kind of a conceptual model of how this might be related to health in humans. So if you change your gut flora, your gut microbiome, this could potentially increase your gut permeability, which is not a good thing. If you have an increased gut permeability, too much more than normal, that can cause an increase in endotoxin absorption. So some of the bacteria in there could release, endotoxins that can get into your bloodstream. That could cause an endotoxemia. It doesn't have to be a lot over a long time. It could be a really small amount getting in there. That can cause chronic inflammation. And then it's been shown over the last several years that chronic inflammation can lead to things like metabolic diseases and disorder. So this is like stair step from changing gut bacteria all the way up to the metabolic disorder. What could we change in here? What can we do, to stop or to slow this down? And one idea is we got to decrease gut permeability. And then there's these things called short chain fatty acids, which are inside your gut that are produced by some of the bacteria. Butyrate is one example that can actually stop that. And then that would again stop the endotoxemia down the road. So this is kind of one idea. And this is where people, I'm not going to get into probiotics and stuff, but probiotic supplements potentially can increase butyrate. Changing your diet can increase butyrate. Exercise could potentially increase butyrate. So this could be, kind of a new field just to stop that metabolic disorder at the end. So this is one of our previous students, Brian Dirk, who did this for his master's thesis. And he was interested in, is the gut microbiome composition different in people that are fit versus unfit people? So we got a group of people that come into the lab. And this was a collaboration with you all at Georgia Southern, as well in our biology department, did some of the work on this. We just got 40 young, healthy adults from around campus, relatively healthy, but they had different fitness levels, different nutrition levels, and they were different body sizes. Tried to get a range, but it was, average 25 years old. And we looked at the phylum level. So if you remember from Bio 101, you got domain, kingdom, all the way down to your genus and species. Well, now we're starting to look at things like genus and species, but this was just kind of a phylum level, big picture, of what's going on in your gut. What we found was we looked at this thing called the F to B ratio, which is a ratio of two of these phylum. And over time, or within this group, we looked at VO2 max. So is everybody familiar with VO2 max, or maximal aerobic output, how much oxygen you can use? And this is a relative milliliters per kilogram per minute. We had people down in the 20s, all the way up to almost 70. So huge range. I'm one of these dots, but I'm somewhere in the middle. Nothing crazy for my VO2. And we looked at that. We found that there was a correlation. And this doesn't look that strong, but it actually is kind of a strong correlation in physiology. We found that aerobic fitness explained about 22% of the variance in gut microbiome composition. So if you knew somebody's VO2, you could basically 22% of that explanation is from their F to B ratio. So the difference in their microbiome basically might predict their fitness level, or is this kind of the chicken and the egg thing what we're working on? Was it these people had this microbiome to begin with, or did it change with their fitness or exercise? We also looked at things like, again, their body size, nutrition, and all that. This was still the biggest predictor. VO2 max was the biggest predictor of their microbiome composition of everything we looked at in the study. All right, so then we took it to the next level, and we got a case study on an ultra marathon runner. See, I do a lot of case studies on crazy people. People call me the king of case studies. I don't know if that's good or bad. Probably bad. But we're adding on to this, and we're going to start doing more. But we got a hold of an ultra marathon runner who was world class. And here's the career highlights. You don't have to read all those, but just know that it's pretty legit. And we looked at that F to B ratio along with the genus and species level, which I'll share in a minute. And if you look at this, if we were to plot this on our F to B ratio of these average healthy adults, we've got to jump up. And this is where this individual will be 7 times higher F to B ratio than the average adult at resting. And so, obviously not that many people are running 100-mile races for fun. So is this, again, chicken or the egg? Was it born like this, or did it change over time with exercise? So then getting down into that genus level, getting down a little more, and there's, again, thousands, probably millions of different types of bacteria that are around and that are inside of you. But if we just get down to here, the top 5 of the 288 genuses that were ranked were these 5. And if we look at what these 5 are, the Glaudia, carbohydrate metabolizer, which makes sense, right? You got to metabolize carbohydrates when you're running 100 miles or whatever. And then butyrate producers. So remember, butyrate is that short chain fatty acid that can protect against that leaky gut. And imagine if anybody's run long distances, it's not that easy on your gut, even independent of eating. But these 3 bacteria specifically might help protect against that. So those are butyrate producers. So again, another connection between health. So then again, we started doing this where we wanted to track before and after training. So this was our kind of case study where we looked at the Western States race, which is just inland of me in Northern California. This is intense. Anybody heard of this? Western States. So 100-mile race. Goes from Lake Tahoe down to Auburn, California, and it goes up past about 11,000 feet and then back down. So it's through the mountains. There's snow there. Even in the summer, there might be snow. So it's a really intense race. We followed our runner here. You might know this guy here, and this is my rental car. So we were all painted up, driving around collecting data. But we collected data on this individual before and after, and some stuff during the race as well. This was the paper we published pretty recently. Again, this is kind of a lot of data, but we got baseline, pre-race, post-race, and recovery gut microbiome. And if you look, this breakdown here between pre-race and post-race, just a couple ones I want to highlight. This yellow one's called Veonella, and that is a bacteria that actually metabolizes lactate. You produce lactate during exercise, long duration or high intensity exercise. This could be related to performance. Then there's another bad bacteria. This one's Haemophilus. There's several types of bad bacteria that could cause disease and stuff. But then you notice that recovery, those went back almost to baseline. So this is really transient. Stuff changes really, really fast. And this has led us to a lot of different questions. So we just got funded. I mean, last night, midnight? Found out we got some funding to do this on a larger sample size, same race. Over the next couple of years, we're going to be going out there. We're going to look also at men and women, different age groups. So we've got to figure out what's going on here. So we're going to look at this in different sample size, Also, the influence of nutrition. I'm working on a study with Rutgers in New Jersey looking at high protein, high fat diets on this. And then better tech. We've been talking, there's so much data here. We're going to need some kind of artificial intelligence program to run through this data and find stuff that we're not seeing. Because every time I talk to somebody, they're like, oh, that's a new bacteria I never heard of. These are people that study bacteria. So we've got to get in deeper with this, you know, potentially use artificial intelligence over the next few years, programs to figure out what's going on. Any questions on that microbiome?
You go with your scatter plot of the VO2.
Yeah.
Sorry.
No problem. Only a couple slides back.
So with the inclusion of the outlier up top, it would indicate to me that this is maybe a non-linear relationship. Do you think that activity level, which is a little bit broader, given that he's running such great distances, that may be a greater driver of this relationship as opposed to VO2?
Yeah, that's a good point. So with VO2, we didn't know if it's their actual fitness or, you know, we also looked at activity level in this group.
Yeah.
It wasn't that crazy. So I think you're probably right. The activity level is going to probably, you know, make that a curvilinear relationship, but we need more data. I mean, what do you think, Greg? Activity level or actual fitness?
Probably activity level, yeah. And also, yeah, and the diet that accompanies the activity level, yeah, it's multifaceted for sure.
Yeah. No, yeah, that's a really good point. Yeah, it's, you know, there's so many things that we can look at, but just in this group of us, our activity level was not, you know, we weren't training right 20 hours a week or whatever. Yeah, that's a good point.
But so, and I know you know this, but if you put activity level on the horizontal axis, it would be lined up a little bit better in that linear type.
Yeah. Yeah, and you know, again, this was self-reported activity level with this group, the 40 people that we had, and you know, we didn't see really anything with that on that. But if we were to add some elite athletes, I'm sure that would.
So there was nothing.
No, not activity. The only thing that was really close in diet was fiber content. What else was close? Caffeine, maybe? Or alcohol, one of those two.
I think it was caffeine.
Yeah.
Yeah.
So the diet, it wasn't even macronutrients, it was fiber.
We probably need to pair some actigraphy with it, right?
Yeah, we should have them, you know, we got some more activity tracking. Yeah.
All right, sorry. Other than, so clearly there are all sorts of implications to your gut microbiome. Is it, so, and it obviously affects gut permeability. Are there potentially other things that it does, or metabolic changes in some level? I mean, I guess.
Yeah, I think this, like, one highlight here was the gut permeability, but definitely metabolism of food, of what you're eating. If you don't have the right bacteria in there, then you have to process that. Yeah, definitely.
Excuse my ignorance, but how exactly do you, I guess, quantify the amount of just gut bacteria? Like, how do you do that before and after a race? Is that something you can do quickly, or is that something you have to like, you have, yeah, like,
Lots of money.
Yeah, and your point is good. We're not even measuring the quantity, we're measuring the types. So we're it's a 16S RNA sequencing, and that can tell you what type are in there, but we're not able to say, well, you've got a pound of this and a pound of that. That would be pretty detailed right there. Yeah.
Yeah, determining absolute changes is very tough to do. One of the things we are doing in the follow-up that we're gonna do next is, Jimmy was talking about butyrate, right? And how the butyrate producers increase. But truth is, we don't know if those butyrate producers increase or dead or alive. We just know the genetic material is there. And so in this follow-up study, we're complementing the microbiome analyses with metabolomics analyses. So we're actually gonna be able to look at, really provides Jimmy and I have been laughing about this, because it's a very catchy word, the metabolic footprint of the bacteria that are in the gut, right? So not just who's there, but what are they doing? So we're gonna be able to get a more robust snapshot as to what's going on.
Yeah.
Yeah, for your ultra runner, pre-post their 100-mile race, did you by chance like measure any endotoxin levels in the blood? Because the idea, if he has an increased butyrate level, his gut integrity may be a little better. He's gonna accumulate less like LPS in the blood, things like that.
We didn't, we were going to, think that was part of it.
Yeah, so we ordered some cartridges. And our French friend didn't get them on time.
Yeah, actually, the day before, I was trying to figure out where to pick them up, and they weren't until like 2 days later.
Oh, that sucks.
I'm
But
kidding. I was pulling my hair out.
if you go to look at the posters from ACSM last year, I was so bummed too. ACSM was supposed to be live in San Francisco. We had like 10 posters. They were all online. Still good because they're always online, but we did a few different studies and looked at things like urinalysis and stuff. And
Yeah.
there was, blood and urine, all kinds. It's not good for your body to run 100 miles an
No,
hour.
I didn't think so, but like, obviously, since he has that increased butyrate, he's gotta be somewhat more protected than the average individual.
I would assume, and it's probably a training adaptation, right? And then look how fast they come back down to close to base. So, yeah, that's good points.
So is leaky gut a condition that can actually be like tested and evaluated, or is it sort of a theory as to the negative effect of like bacteria, gut bacteria don't want? I know in like nutrition right now, it's always sort of a battleground of people who think they know what they're talking about by kind of reading the literature and then spouting off. But I've heard the phrase leaky gut a lot from people who are like, eat this to stop your leaky gut and everything.
Yeah, I don't know if they would be able to tell that from a quick test, but you can, if you go to the doctor and they can look at your lining of your intestines as well. You probably do some kind of scope and stuff, and then I don't know, blood markers, right?
Yeah, you can measure the
Yeah.
LPS in
The
circulation.
bacteria is only going to get there.
It's very hard to measure.
Yeah. Yeah, so that'd be the indirect way to do it. So, yeah. Good on that. All right, so we looked inside the body, outside the body. Now let's look forward. So this is something that I did not even know existed until like 2017. We were approached. I just got to San Francisco State a couple years before, trying to build my muscle lab. I was approached by a guy named Aaron Stanton who worked in tech. At the time, he worked at Apple. He invented iBook. You all have Apple and you use that app. He invented that, and so he's a pretty big programmer. And he was actually, pretty out of shape and unfit when he was working, obviously on the computer all day. I started picking up the Oculus Rift at the time. I had a VR headset playing games. And he noticed his heart rate was going up while he was playing these games. He's like, I can't tell if it's, he didn't say it, but it's parasympathetic response. I can't tell if I'm scared or if it's actual exercise. Come in the lab, we can tell you if it's exercise or not. So I'll talk about here, we hooked him up to a metabolic cart, and we found out that he was actually exercising. It was increasing his VO2 at the time, and he was using calories. This is kind of interesting. And so, you might have seen, everybody probably played a Wii, Nintendo Wii back in the day, right? That was, a lot of people were studying that. This is the new thing to change fitness. Problem with Wii is it's one company, and they only had a handful of games. Virtual reality headsets have been out now for about a decade, these relatively good ones. And there's 10, 12 games coming out a week on these. And they're on Steam. If anybody plays computer games, it's all on Steam, or you can get it through even Facebook now. So it's just everywhere. Anyway, kind of the background of this story is we started getting into studying the impact of virtual reality gaming on health and fitness in different populations. So has anybody ever used a VR or AR headset? Yeah. What kind do you use?
I have the Rift S.
The Rift S? Okay, cool.
I'm actually not sure which one I would use.
Okay. The hand controllers, cool. Yeah, it's probably the Oculus Quest or the Rift, or HTC Vive is the one that we use a lot, which is really cool. Now they're cordless. A lot of them are cordless, pretty high resolution stuff. So they're getting pretty cool. Yeah, so we started working on this. I'm going to just play the video so you can see. This is one of our students in the lab playing this, hooked up to a metabolic cart. When I saw Aaron playing that, I was like, I'm pretty sure you're exercising. I don't think this is a parasympathetic response. So, yeah, this particular game, I don't know what that one's called, but it's where you actually have to hit these things flying at you. You can bounce them away and use your hand as a shield as well. But yeah, so pretty intense. And you can see that she was hooked up to a metabolic cart there. So just kind of backtracking, talking about technology, kind of tying it all back together with technology again, is that, we've been measuring VO2, or people have been measuring since the mid 1850s. So they didn't know what VO2 max was necessarily, but they were measuring oxygen consumption at that time. This was considered portable. This table, they weren't measuring CO2 or anything like that. And this was the first time that we had hooked up one of our students, Dulce Gomez, to our Cosmed, that cart. This was the old lab before we renovated, but this is when we started seeing like, wow, this is actually looking like it's exercise. Just a quick, I know we're running out of time here, but a quick backtrack just to look at the technology advances in that. The first real portable system people will say is probably in 1906. Carl Zeiss, and this, I guess, a hookah-looking device here, where you have a backpack or bagpipes or something, where you could actually be portable, measuring gas exchange. Up to the first commercially available one was around World War II. A lot of technology was coming out at that point. And then if you remember what happened in the 1950s, this is when actual microprocessors. So instead of having big tube computers, started making microprocessors. This is like the start of the Silicon Valley, start of internet came out about a decade later or so. But this was, again, a more portable device. And then up until the 80s, 90s, they started, Cosmed is that company, they started making the K units. Now they're on what, K6. So it just keeps getting better, more portable, still pretty expensive. It's not cheap, but we can do this portably. In the lab, we still typically use our Cosmed metabolic cart here. It does have a limitation on the lines, so you can't move too far away, but it still works pretty good in the lab. And if you want to see some of the work we've been doing, just check out vrhealth.institute. We actually, I'll show you in a second, but we rate games based on caloric expenditure. And then to make it easier for the general public, we do that in kcal per minute, so they can see potentially how many calories they consume per minute. So the first study we published on this was 2018, and this was the first study that I know of that used virtual reality equipment to look at basically metabolic cost. And we did this, again, VO2, relative VO2 to your body weight. Right now, everybody's probably a little above 3.5 mLs per kg per minute should be resting. Maybe if you've been moving a little bit higher. So we got rest on a group of people. Then we had them play, and these were randomized as well, so they weren't in this order necessarily, but we had them play a game called Audio Shield, kind of like blocking different colored things coming at you, rhythm game. We found that was you use about 10 to 13 calories per minute while playing that. So, relatively moderate physical activity there. Then we had them play an archery simulating game where this one's pretty fun, where you get to kind of like Legolas, shoot things coming at you and you have to dodge them, everything. And this was even higher. We were over 20 for their relative VO2. And then one of the most intense games I've still ever played is this boxing game called Thrill of the Fight. And it was 15 plus calories per minute. So pretty, I don't know how many minutes to play that for, but it was pretty vigorous exercise. So it does cause exercise, right? So that's kind of like a given, but we were trying to break through the mold to say that gaming can be exercised. One of the more interesting things we found out, so we had, light, moderate, and vigorous activity. We also asked RPE. So you're all familiar with rating of perceived exertion. How hard do you feel like you're working? The Borg scale. So on a treadmill, we did a max test as well. So we got, data on them for where they were at light, moderate, and vigorous on the Borg scale. And then when we asked them right at the end of their gameplay how they felt they were doing, it was consistently lower. So they were light, and moderate for their RPE, saying that there might be some disconnect, at least in the short term, of the psychology of these people feel like they're actually exercising less than they are in real life. This would probably be the same thing you would do if you're playing sports, playing basketball or soccer. You probably rate your RPE lower than what it really is because you're having so much fun. So now we're starting to look at things like fun, motivation, cognition. Not a sports psychologist or exercise psychologist. That's why we're having people work with us. But it's, this is like the next level. It's how can we get people to play these longer and have fun doing it? So, like I said, we're kind of working on that now. This was at the ACSM, Southwest ACSM. I think we also did it for regular national ACSM. But just looking at RPE, it was consistently lower than estimated RPE of what it should be in both novice and experienced gamers. So maybe we thought, maybe their game experience would change this, but nope, it didn't matter about your experience at all. And this was one of Trenton, one of our other grad students' thesis we did a little while ago. So yeah, it is a valid form of exercise. These are some of the most popular ones. Anybody heard of Beat Saber? That's probably like over the last 2 years, at least been one of the most popular, but Box VR. Supernatural is kind of like an online personal training kind of thing. They're all pretty fun. Yeah, so we're going to continue studying these things. There are some barriers though, like you can imagine, besides the cost. So I talked about tech equity, the cost is there. It's getting cheaper, but not everybody can go buy a $300 headset, right? So that they're going to become, cheaper and cheaper as time goes on. but also gaming culture. Gamifying fitness requires people to get up and move around even if they're in their house. So most gamers have really comfortable chairs and they kick back, they're gaming all day. This requires you to get up and move around a little bit. Accessibility, again, cost, equity, and then engineering is kind of the stuff that we're working on with these companies, hardware and software. So I'm not a game developer or designer, but I can say, hey, if you want to make this better exercise, probably should build it like a HIIT workout. Hey, if you want to make this more fun, maybe you should do this that lowers RPE. So we're doing stuff like that. We're working on things like motion sickness, improving fitness tracking with artificial intelligence, and then designing different modes of those. So I'll do a quick recap of a study we're working on this. Anybody get motion sickness regularly? Right? In the car?
Only when you're driving across the country.
Yeah, motion sickness is kind of a problem with, not everybody, but 1 or 2 out of 10 people will get motion sickness. And we're trying to figure out why that happens and what we can do to stop it. This was a study done by Angelina Wong, where we looked at, and this is me on a VR bike, which is pretty cool. The bike actually moves while you're riding on it, kind of like a Peloton, but you're in VR. We did it like with that and with also basically riding the bike, but watching the video in front of you. So is there a difference between having the screen this far away or this far away? Not significantly. So this just shows pre and post cycle and pre and post cycle without the VR headset. There's increases in things like nausea, disorientation, and total severity, but it wasn't different than if you're just looking at it on the screen. So we're going to dig more into this. This was with a small sample size, but looking at why some people get motion sickness and some people don't. So what's the best way to track fitness? So probably a lot of people have heart rate monitors in here. That's one way to track fitness. You can also measure movement displacement with these headsets and hand controllers. So we tried to look at between actually hooking somebody up to the Met cart versus movement and heart rate, which one's estimated kcals the same. So with movement and VO2, that was pretty close. Surprisingly though, I used heart rate to measure my, to basically track my fitness, and heart rate overestimated kcals significantly almost in everybody. This was kind of crazy because we were using heart rate as our main measure without these two. And again, if you look at this, this is just a plot difference showing that as you get to higher heart rates, so this is the mean heart rate, it gets even more off on the estimation. So probably would expect that as well. But what we found is we probably want to combine these two and also come up with new heart rate algorithms in VR gaming specifically to estimate caloric expenditure. We're working with some companies now to do that. And one thing we've got is an app on your phone. So if you guys have access to a VR headset and you want to play some of the games you have, you can download this on your phone if you have a heart rate monitor, wristwatch, or chest strap. This is still in development, but you can get the beta version and try to estimate your kcals while you're playing. And then the more data that we get from that, the better these algorithms will get. So what's next for this? Again, the cool thing is there's endless opportunities. There's developers around the whole world making games every day, probably new games coming out. Doesn't mean they're all going to be good, but they're coming out. And so you can have endless opportunities to gamify your fitness. There's new modes of VR, and I didn't even mention augmented reality, but that's basically what's going to be coming out in the next few years, where you'll be able to see the world, but you'll be able to see like basically a hologram in front of you. Like the closest thing I can think of that most of you have done is you play Pokemon Go with your phone. You're kind of using augmented reality, seeing a little Pokemon jump around. It'll be something like that. So there'll be new modes. A couple of things that we're working on is this is called a Flicer. There's a few companies that make this, but it's a thing you lay down and you can move with your arms. So it's more of an upper body resistance training device. It's got cables that you can change, these little rubber bands to change the resistance. And then we've also got those bikes I showed a little bit. Those are from Boarding Track. They're basically a Peloton, which instead of a screen, you've got goggles that you wear. And then again, the next thing is responsive exercise. So the thing that we're working on and a lot of people are working on too, with that game Supernatural, is if we do a fitness test on you in VR, we could build a training program based on your goals. You want to lose body fat, you want to have better cardiorespiratory fitness, more muscular strength. We can go through that and basically have an AI help build that. So not necessarily putting your personal trainers out of business, but personal trainers might be using and helping AI to help that client work on that kind of thing. So we're working on that as well. Okay. So any questions on that before I do the last little bit here? We'll If anybody wants to get involved in this, all you need is a Met cart. I can hook you up and you can do some studies here as well, even multi-site studies. Pretty cool stuff. Though. Okay, last couple of slides. So again, just showing this, I want to show you just some of the technologies that I talked about, the microbiome reference, the CNIA, the VR headset. All these have come out over the last basically 100 years, but it's only now that they're converging and being able to be used together for something like exercise physiology research. And then this kind of builds on that integrated view of exercise physiology. You know, with muscle physiology, our idea is just to understand what's going on in the muscles so we can help make them work better. Gut microbiome, understand the gut microbiome's influence on both health and performance, and so they can work better. And then exercise gaming, so developing really effective, sustainable, and then really important accessible devices and games for improving health. So thank you all again for having me. If you have any questions, my contact info is up here.
Great talk, Jimmy. If anybody has any questions?